Hello everyone. So today we're going to be discussing genetically modified organisms in this lab. The first topic that I want to introduce you all to is a problem that we encountered as a society, something like 100 years ago and for a long time in the past as well. This is the problem of diabetes. Diabetes is a disease in which humans are unable to produce productive insulin. Insulin is a hormone that is made in the pancreas that is produced by the pancreas when you have a lot of glucose floating around in your blood. Now, from our other lectures, you all should know that glucose is a sugar molecule, and when there is a lot of this sugar floating around in the blood, it poses a risk to kidneys, because in the kidneys, blood is getting squeezed through tiny capillaries, and when you have sharp sugar molecules in high abundance squeezed through tiny pipes in the kidneys, they actually can cut them. And so one of the symptoms that we see in diabetics is that glucose actually escapes into the urine. That's bad. Eventually, enough of that glucose will cause perforations or holes in your tubes in your kidneys, and you will start losing blood through your urine. And while you might intuitively not know that sugar in your urine is bad, if you are peeing out blood, you certainly know that that is not a good sign. Now, diabetics, there are two different kinds. Those that are genetically diabetic, type 1 diabetes, and type 2 diabetes, some sort of environmental induction. For the purposes of today's lab, we won't really talk about that, but we will focus on the fact that neither of these two have a working copy of insulin so that they cannot regulate how much sugar is in their bloodstream. In order to survive for a long time with this disease, we needed to figure out a way to add insulin to individuals so that way their blood sugar did not spike. Now, we could potentially take it from humans, but this requires mashing up the pancreas and extracting the insulin. Now, I don't think it's a great idea to extract it from living humans, and we can't take out the pancreas. It's not a regenerating organ, so we need to find another source. Well, fortunately, pigs are similar enough to humans that we could take insulin from a pig pancreas and inject it into a human, and you'd have that same effect. It puts the sugar that's floating around in the bloodstream into the red blood cells, so that way it's not going to get to the kidneys and cause cuts. But I don't need to tell you that there's only so many pigs on planet Earth, and diabetics need insulin regularly. So we could not produce enough insulin from pig pancreas extractions in order to address the problems with diabetes. Therefore, we had to find another way to produce it. And this method would instead take insulin production from mammals, like pigs, and put it into a bacteria, like E. coli. We were obligated to transgenically modify E. coli, put insulin into it, and have E. coli produce that insulin that we can then harvest and give to diabetics for them to be able to hide glucose in their red blood cells. Let's go to the whiteboard and talk about that process now. So here we'll pick up where we left off by talking about how we use E. coli, a bacteria that'll be illustrated in orange in this diagram, in order to produce insulin. Well, we know that originally E. coli does not have the capability to produce insulin. It's a bacteria after all, and insulin is a hormone found in animals. But E. coli has a circular genome, and it does not distinguish between its circular genome and other pieces of circular DNA. This allows E. coli to uptake things called plasmids. So here we have a circular band of DNA that we call a plasmid with an EcoR1 cut site. Now, if we can get this plasmid into E. coli, it will make whatever genes are present on that plasmid. So, we can take our target gene from humans. This is our insulin gene. And if we use similar sticky ends, the cut sites, on both of these molecules, you will then get a plasmid that has our target gene of insulin inserted into it. EcoR1 is called a restriction enzyme or a cutting enzyme. And so it just cuts both of these pieces of DNA so that way they recognize each other, and some percentage of the time, if they happen to be floating next to one another, they bind, they connect, they ligate. And in this way, you're able to take a gene from humans and put it into a bacterial genome. But now we face the problem of this plasmid with insulin, being floating away and outside of E. coli. If we want it to be expressed in E. coli, we have to get it into the bacteria itself. And this is what we'll be replicating with our kind of silly exercise today. In order to get that plasmid into E. coli, we have to temporarily create holes in the E. coli. The way in which we do this is kind of violent. We put the E. coli in a hot bath, literally boiling water for something like 30 seconds. Now, most of the time, if you put bacteria in boiling water, it kills it. But if you put it in there for a very short period of time, the process of dying begins to happen. The E. coli will begin to expand, but it creates all of these holes in it. If the plasmid is floating near the perforated or opened up E. coli, some percentage of the time it will float in, and some percentage of the time it won't. If you want to save those E. coli that now have a plasmid in them, you can throw that E. coli from the hot bath into an ice bath. And you'll be replicating this by opening and closing the E. coli with our exercise. Now, those E. coli that have the plasmid can then be grown out on a plate of sugar media. Because bacteria, we all know, does great when it's got a bunch of food on any left out food surface. Just go ahead and try to leave a gallon of milk on the countertop for a day. Lots of bacteria will love that warm, nutrient-rich environment and begin to replicate. But we don't want all of the bacteria to grow. We want just the bacteria with the plasmid. And that's where this green strip of the plasmid matters. That green component of the plasmid carries antibiotic resistance. And if you put antibiotics, which are designed to kill bacteria, on this plate, the only bacteria that will grow are those bacteria that have the plasmid, because they carry both antibiotic resistance and the insulin gene. Then, by picking up, and literally you use a small little spoon to pick them up off the plate, by picking up these bacteria colonies that grow and culturing them further, you can proliferate or copy these bacteria, and then they will go ahead and create insulin. And in this way, we take a bacterial genome and a gene from humans. Now, because humans are not the same species as bacteria, we call this gene a transgene. And by putting in a transgene into our bacteria, we create a genetically modified organism. And this E. coli that now carries insulin does not know that it is a human gene. It can't tell the difference. After all, you know that it is just a whole bunch of A, T, G, and C. It's going to make that gene without asking any questions. And it's going to secrete that insulin out of the bacterial cell. So that way, if you have a giant barrel of these bacteria growing in something we call a bioreactor, you can then take the liquid that they're growing in and purify the insulin out. And this is the method in which a vast majority of insulin produced across the world is made for diabetics. Our insulin production is transgenic. So let's go ahead now and try to replicate this process of heat shocking and then cooling bacteria down to see the success rate of this transgenic introduction.
Okay. All right, and this is our visual representation of how E. coli will be taking plasmids, or if they will take the plasmid. So this, our spiky ball, is representing our untransformed
E. coli bacteria.
E. coli bacteria in this case, for the insulin production. And the paper ball that Paul has is going to be our plasmid that has our transgene in it. So this is a process of transformation if our bacteria takes the plasmid.
Successful transformation looks like the paper ball being caught into the bacteria staying alive. But as you'll find out today, it can be kind of difficult to do. So what are we going to do to replicate this process, Becca?
So in order to replicate this process, we're going to do 3 different tests. So the first one, I'm just going to be trying to get a feel for how to catch the plasmid in our bacteria. And Paul is going to be throwing it at me. I will have my eyes open, and I'm just going to try and catch it.
And now, Becca, what happens what are we replicating when you open the ball versus when you close it?
So upon opening the ball, as Paul was talking about earlier, this would be the hot bath. So when the bacteria is in a hot bath, it begins to break down the membrane of the E. coli, causing it to have perforations or holes, which is the cell practically beginning to die.
But in those expansions, you allow for the plasmid to sometimes float in, and then how do we close those holes back up, Becca?
So the closing of it is the opposite of a hot bath. We dump it right into a cold bath, and it will shrink the cells back up and close all of the holes in the membrane.
And if the plasmid got in there, the bacteria doesn't know that it wasn't there from the beginning, and we've got a transformed piece of bacteria. So let's model this process. I'm going to go ahead and yell hot at you to open it, and then cold when it's getting close. Here we go. Y'all will be doing this 10 times to get an average rate. We'll just do it twice. Hot, cold. Oh, close, close, close. Not quite, a failure. Let's try it again. Hot, cold. Whoa, look at that. That wasn't scripted. That's good. Look at that talent. So we'd have a 50% success rate. But what's different about this process compared to when E. coli is trying to do it?
Well, Paul, we have eyes, which make it a heck of a lot easier to see when that is coming at me to close the bacteria.
So how can we make this more realistic, Becca?
Well, bacteria don't have eyes, so I'll just close mine.
All right, let's try it again with eyes closed. Ready? Hot, cold. Nope. Round 2. Hot, cold. Oh, we got close, but not success. Again, y'all need to do this 10 times to get an average. But that's even too generous still. If you do this, in the past, I've seen success rates of up to 10%. Now that's not anywhere realistic to what we see in actual E. coli. E. coli actually have a success rate of something like 1 in 10,000 to 1 in 100,000. So how can we make this more realistic, Becca?
Well, I can turn around and do it.
Let's give it a whirl. Hot, cold, up, and that is how you hit a student in the head with a paper ball. One more time, for comedy's sake. Hot, cold, not working. But why does this actually work? If it's so rare, Becca, how can we actually get transformation events in our vials?
Well, since there is a chance that it's going to happen, we know that it will happen eventually, so if we do it over and over and over and over again, eventually we'll get enough of it that we'll be able to do something with it.
So how many bacteria could you fit in a little vial like that?
A very large amount.
Exactly. So even though this is a rare process, we rely on the game of large numbers. And by doing this process hundreds of millions of times in each little vial that we're running it, because that's how many bacteria can fit in one of those vials, we will get some transformation events, and we can select those transformation events on our plates. Ended. All right, so here we are with section 2 about targeted genetic additions. In order to discuss this section, we first have to start with our template DNA. And so y'all know that DNA is made of 4 bases. You've got your A's pairing with your T's, your adenine with your thymine, and your G's, guanine, pairing with C's, cytosine. Now, in this sequence, we've got both strands that are complementary, but really we're going to be paying attention to just the red one here. Because what we want to do is find a way to insert different sequences into this strand of DNA. There are 2 ways that we can go about this. The first uses pattern matching with something called transposons. Now, transposons are limited in the fact that they require pattern matching, and they always require something called inverted repeats. They use inverted repeats that essentially are the same thing, flipped around, read backwards like palindromes, and then they have a sequence in the middle, and whatever's in the middle gets inserted into the DNA. So looking at this sequence here like you have in your lab, you see that the front of the repeat reads TTACG. Going backwards on the other side, it's also TTACG. There's our inverted repeat. Now, whatever is in between those 2 repeats is going to get shoved into the DNA. And in lots of cases, you can have over 5,000 letters in there. And just those 5 will drive the insertion of the 5,000 as long as you have the transposase enzyme. So here's our sequence. Let's see if you can spot where this would insert on our template DNA. I see here TTACG, followed by a GCATT. So here's our back half of the repeat, front half of the repeat. This means that this transposon would insert 5,000 or more new bases right into the sequence. So this thing would be super long. Transposons therefore do these kind of genetic additions to your sequence. But there's a new way, something that's been developed in the last few years, called CRISPR technology. Now CRISPR operates much more like scissors that can cut and paste. Rather than doing these giant insertions, CRISPR can take a sequence and replace it. So for example, you can design CRISPR enzymes that will find the CAAGA and replace it with a different allele here, CTTGA. So can we see that somewhere in here? Yeah, right here. With the CRISPR-Cas9 system, this sequence will be removed and be replaced with a CTTGA. And in this way, this is an allelic replacement, and this is a targeted addition. But are both of these transgenic? Well, yeah, because this inserts genes from something else, bases from somewhere else, oftentimes by design of the biologist, though transposons occur in nature, and humans have a history of transposon insertion as well. Later on, you'll be reading about this type of process naturally occurring in one of your foods. CRISPR is actually a mechanism of bacterial immunity that we have hijacked to do something that we want it to. But both of them are genetic modifications and therefore transgenic, therefore producing genetically modified organisms.
All right, now we need to do some reading. So what you need to do is look up the wiki for genetically modified crops, and you're going to do a little reading on it. You need to focus on the history and look at breeding and how breeding is different from genetic modification. Focus on some newer updates, I guess you could say, to how genetic modification is done with plasmids and restriction enzymes, and then check out some of the modern uses and how that is being used today. The second section, check out methods and focus on agrobacterium. That's the one that we covered today, with the type being transgenics and how that works.
And which particular transgenic are they going to be paying attention to?
We want them to focus on BT.
And what's BT, Becca?
BT is a name that is derived from a bacteria called Bacillus thuringiensis. And this is most commonly used today in corn, especially.
All crop plants.
Well, and lots of different crop plants.
Or lots of different crop plants. Sorry, not all. Not all crops are, oops.
But many.
Yep.
Typically it's found in corn. And what this does is, Bacillus thuringiensis is a toxin to insects, however not to humans. So when this is in our crops, it kills the insects that try and eat them, but it's completely safe for us.
And last but not least, has any kind of agrobacterium-mediated transgenic event happened in any foods that we might eat that hasn't been done by humans?
Yeah, a very, very, very long time ago, it happened in sweet potatoes. So I would also like you to go look up the sweet potato wiki and do a little reading on that. It's pretty interesting.
Cool. And we'll see you when you're done with that reading. All right, so last part of the lab. To make sure that we understand what are transgenic organisms, what are genetically modified organisms, and what are not, I've got a little pop quiz for you. Which of these are genetically modified? First, we want to talk about dogs. Now, dogs are domesticated from wolves. We know that they share ancestry. I think it's fairly clear, right? Don't you see the aggression and wolfiness of these teeth? I mean, look at those jaws, right? Now, Odie here is part husky, part lab, and part pit bull. He has a mutation that he's very generously showing us, where he's got a white patch on his chest and white patch on his paws. This is a mutation that occurs within dogs that changes their coloration from completely black to this white patchiness. Is Odie an example of a genetically modified organism, being this mix of husky with this mutation for coat color, Becca?
I don't think so, Paul.
Exactly. Rather than being a genetically modified organism, Odie is the product of selective breeding. We have found these mutations naturally occurring in dog populations, whether it be in huskies or labs, and this particular one occurs within labs, so he gets it from his lab parent. And now, because his previous owners wanted that, or the previous breeder wanted that, they selected to breed those two different varieties together and produce this magnificent, wild, ferocious beast. This is an example of classic breeding. We can choose which things we want to be parents, which alleles we want to be passed down, and create the very cute, adorable products of them. Breeding is not genetic modification. For our next example, we need to turn to another organism with a spine, fish. And in particular, we're looking at glowfish. So here we have an image of some glowfish that have the ability to glow in the dark because they have a gene from jellyfish. Is this different color pattern an example of a transgenic organism, Becca?
Pretty sure it would be. You said it came from jellyfish.
Exactly. Because we moved the gene from glow-in-the-dark jellyfish into regular fish, and they look kind of cool, right? That is an example of a transgenic event. You're moving something from different organisms. In Odie's case, that gene occurs naturally within dog populations. Here he comes. As the gene is found within different dog populations, it is not transgenic. It's naturally occurring, and we can breed for it. In the case of our fish, it's not naturally occurring. It comes from jellyfish, and we've transgenically modified it. For our next example, we need to talk about strawberries, the fruit crop that's most often associated with genetic modification. Because if you look at different strawberries, the wild strawberry is incredibly small. The strawberry that you get at the grocery store is going to be incredibly large. The difference between these strawberries is that the wild strawberry is diploid. It has 2 copies of its genome. The strawberry that you get at the grocery store, oftentimes grown by Driscolls, and oftentimes those patents come from UC Davis, where I did my PhD. No small part of my PhD was paid off of strawberry patent money. Thank you, strawberries. Thank you all for buying strawberries, supporting graduate student education. These strawberries just have 8 copies of their genome. Nothing else is different. Are strawberries, therefore, an example of genetically modified organisms?
No, it's just strawberry.
By virtue of the fact that you just increase the copy number of the genome and are not introducing a gene from something else, these are not genetically modified organisms. They are the result of selective breeding. We found a diploid strawberry that then naturally mutated into a tetraploid strawberry, meaning it kept an extra copy of its genome. And from the tetraploid strawberry, we then looked again for a octopoid strawberry, copying its genome again. These are natural variants that occur within the wild, and we just happen to use them because they grow bigger fruit, more for the farmer to sell. Breeding versus transgenic events versus breeding in our wolves. Our last example are soybeans, the crop that has Bacillus thuringiensis gene for the toxin that kills insects. Are soybeans with BT examples of transgenic organisms or genetically modified organisms, Becca?
Yes, they are.
Yes, indeed, because the gene derives from not soy, they're an example of a genetically modified organism with a trans gene. You can always remember that because trans means across, and in order to get the gene into soy, you had to cross or go across the family tree of living things, go from plants to bacteria. So hopefully this helps you discern what is truly GMO versus what is truly GMO versus what is done by breeding. And I'm going to go ahead and tell you that these misconceptions about genetically modified organisms are incredibly common. Once at the doctor's office, we were asked, are we allergic to GMO strawberries? There is no such thing as GMO strawberries. They're not sold. We have that breeding process to breed for octoploid strawberries, but they do not have trans genes. And this was coming from a doctor. So make sure that you sound educated and know what you're talking about when discussing genetically modified organisms. They make a big difference in how you decide what you eat and what's growing in our environment.